So did you want to try the Hangout?
It'll have to be on yours. So. If you want me to log in, it's me.
Yeah. It should.
Should we just huddle around him? Yeah, I know.
We might have to.
For the sake of memories. I can log in as me and then take it from there. You know. So you have to. I'll start a Hangout. Anybody remember the word?
You got an invite.
Yeah, there we go. That's about it.
Why don't you start prefacing your
Yeah.
your presentation and then we'll do a quick break.
Yeah. Yeah, so my MacBook got into an argument with the projector and lost. So we were going to see if I could Google Hangout the presentation from the MacBook to this one, just because I have my environment set up for doing some live demo stuff in the second half of the talk. But we'll see if it's worth all the trouble. If it's not going to work, then I can just plug in the thumb drive and play my slides off of this one. And
you need an adapter?
I need a better one than these two.
You're working another error.
Yeah, it's
Particular MacBook error is
yeah, it's
DNC?
Yeah, we're going to see it.
DNC?
huh?
DNC is Google Hangout. That's what they're trying to get working.
Yeah, it's yeah, it's unfortunate I never met Brian Hunter, because we were actually stomping around the University of Chicago around the same time. I was an undergrad and taking David's operating systems course, and picked up his book on Python around the time John Hunter was starting to work on Matplotlib. So we almost were in the same circles, but then I took off for graduate school and worked on environmental microbiology, and in a minute we'll be showing what I worked on, and kind of made the transition from working in Perl and then R, and switched to doing as much as I could in Python and doing all the graphing in Python and saving myself some sanity. Brian, how's it working?
I'm having a hard time with the
Well, I'm gonna start, and
Okay.
it's so like I was saying, and if you've read the preview on the website, I had kind of a two-part talk. One was how I was going to use I was using Matplotlib for surviving graduate school, and the other was how I was using Matplotlib for goofing off when I was finished with grad school. And you'll see in a couple of minutes how I kind of use the same stuff I learned in one arena to have a little more fun in the other arena. So what I was doing was first plotting genomic data from the bacteria that I studied in grad school, and you can see a little one of them in the corner of the screen there. And the other thing I was doing was kind of goofing off with this data that Major League Baseball has on their website that kind of shows the behavior of all the pitches their pitchers have thrown over a few years now. So the first half of the talk, what I was doing in grad school, the lab I joined has been studying this Cyanobacterium Prochlorococcus for now, well, they actually are the ones who discovered it in the late 80s, and they've focused on it ever since. And the reason they found it so interesting, there are several reasons. First of all, it's actually it's a very simple organism. It's most of them are about half a micron across, which is pretty small even by the standards of bacteria. They divide once a day, which is pretty slow compared to the usual lab rats like E. coli that are more like once in a hour or half hour. Most of the genomes we study are less than 2,000 genes, and that, again, even by the standards of bacteria, is pretty tiny. There are simpler bacteria that grow in very specialized environments and have lost much of their ability to kind of sustain themselves, and they live as symbiotes inside of other organisms, so they have smaller genomes. But here we have a bacteria that has a small genome and yet has all the tools it needs to be an oxygenic phototroph. That is, it splits water and fixes carbon dioxide in photosynthesis like a plant chloroplast does. So that means it needs this whole package of genes for encoding photosystem 1 and photosystem 2 and the carbon concentrating and carbon fixing mechanisms. And that takes up a pretty big percentage of the genome. And I'm not sure what yeah, that was displaying better on the other one, but that was just going to be a picture of the let's see if I yeah. That was just a map of the world and showing where the chlorophyll concentration is highest and lowest, and what the point of that is for Prochlorococcus is that Pro actually thrives in the areas of the ocean that are kind of the most difficult to grow in because the nutrient concentrations are lowest. So it's a little ironic that they're most numerous where it's the most difficult to grow, but the important thing there is that there's less competition from other more complicated cyanobacteria or photosynthetic eukaryotes or other things that compete for the same niche. And Prochlorococcus, with its small size and again its simple genome and simple cell architecture in general, has an advantage in surviving in that kind of environment. And yeah,
Do you want to try to hang out?
I'm worried that the other pictures will be as bad as that first one, but I guess it's just that. Well,
Hangout should be inviting you. I don't know if you want to check it. It's like quick for
Well, let me. Yeah, most of these are.
Okay, yeah. Hey, Greg.
Yeah.
Brian has his announcement too.
Oh, okay.
We want to take a quick break.
Yeah.
Because I think it
Yeah.
would be helpful. Okay, so guys, real quick, we're going to break in.
I did it once with basically R and a mix of Python to do the number crunching in the beginning, R to do a little more, R to do really the plotting, and Illustrator to clean things up for presentation because I wasn't happy with the results coming from R. And I got through a whole paper doing it that way, and then picked up Matplotlib and started doing things with Matplotlib, and redid that plot with Matplotlib and had a lot more fun. And the same is true of another figure I have to show you here, and that's plotting that CAN genome on a single genome. So the genome is circular, but we usually plot it as opening it up and laying it flat on the x-axis of the plot, and then plot things on top of that x-axis. And here, what I'm showing you is basically where the genes of the flexible genome, or the genes that are not shared between all Prochlorococcus, are clustered. And what we found for really almost all of the genomes we have sequenced so far, and seeming to hold true for those unassembled samples from the ocean, what we were finding is that those flexible genome genes concentrate in certain parts of the genome. So in this case, this strain is especially prominent example of that because it piles everything up there in the middle of the genome, or middle of the plot anyway. And what we find is those are islands of extreme variability, and they are the kind of the hot spots for relatively rapid genetic change in the Prochlorococcus lineage. And conversely, if you plot those core genes, or the photosynthetic genes, or the cell division genes, they kind of avoid those hot spots, and they're in what are the lower parts of this graph, and don't vary so much between the sequenced examples. Was there a question? Yeah, exactly. So the question was about the iron stress and phosphate stress example I mentioned earlier, and how genes relating to resisting phosphate starvation or surviving iron starvation are found in different parts of the world in Prochlorococcus. And what the questioner was asking is, are those genes then concentrated in these highly variable islands? And the answer is very much yes, and that's been one of the big stories we've kind of developed in the time I was in the lab, and I didn't have so much to do with the iron or phosphate starvation stories, but I had a lot more to do with the story of how different strains tolerate changes in light intensity. And that's actually what I was showing here when I first made this plot. You see in the legend I'm talking about HLI genes. The HLI stands for highlight inducible. And these were genes that were known for some time to be upregulated when these cells are exposed to changes in light intensity or increases in light intensity. And what we found is there were actually different kinds of highlight inducible genes, a core set that are found in all Prochlorococcus and are found in those core regions of the genome I was talking about. And then a phage-like set that seemed to actually be inserted in the genome and carried between cells by the phage, the virus that infects these cells. And those are generally piled up in those more highly variable regions. So that's what the X's and O's are showing you here, and the phage-like ones are especially concentrated in that highly variable region. Yeah?
How many sequences did you prepare to get that? And what kind of software does it use? And how long does it take to get that?
This plot is generated just from those 12 sequence genomes that I talked about that were kind of the first wave we got. And deciding whether a gene has been basically gained rather than lost in the evolution of Prochlorococcus is it took a little while, but I pretty much wrote all that in Python and did that myself, and it, as I recall, it ran in an hour or so, so it wasn't a terribly huge job. And again, I was doing a lot of the initial number crunching in Python, even when I made the first version of this chart and the other chart with a mishmash of other tools. So I had 1 foot in the Python landscape and 1 foot in a very unfriendly landscape. And actually that's what I wanted to talk about next, is kind of my journey through this. What I was doing was doing a lot of number crunching in Python and dumping results to a spreadsheet or a CSV file and re-importing that in R and making a plot in R. And I don't want to trash R too much because it's a fairly nice plotting tool, but I never really could get it to that last 10% of polish, and things like getting the figure legend in the right place and getting the colors the way I wanted them, and getting line widths and everything in the right ranges generally meant touching things up in Illustrator after exporting a PostScript file from R, and that was okay when I did it once, and that's kind of how I fell into it. Then my advisor would have some comments or recommendations, or you need to fix this, and I'd find myself starting over again, doing some of it in Python and doing some of it in R and doing some of it by hand in Illustrator, and generally forgetting where I was. And of course the worst part is when you come back 6 months later and you have a directory full of plots you made somehow. You might even be able to find the Python scripts that you used and the R scripts that you used, and the Illustrator steps, of course, are just mousing around by hand, so you can't put that in version control. And you, to some extent, have to start over every time. So, like I said, I got through, I guess, my first couple of years and put out one paper doing things that way, and then kind of swore I wasn't going to do this way again. So that's when I started really digging into Matplotlib and finding that there was pretty much an option or a setting for every little option that I needed to change. So For example, my advisor was always telling me to make the fonts bigger because it seems like the default font size in every plotting package is too small to read in a PowerPoint presentation, or actually pretty small even in a printed publication. So she was always on the campaign to make your font sizes bigger. And now I could just do that by changing one line and pushing a button and rerunning the script, and avoid Illustrator as much as possible and avoid switching tools as much as possible. And even then I kind of was now having to decide between putting all the options I wanted in either in the Python script that generated these files, and then I could store that script, or write it so it just takes a bunch of command line options and make sure I save a shell script or something with all those command line options. Neither of those was really what I wanted, and the trouble with the shell scripts is then I end up saving all these bash scripts all over the place, and now I'm doing half of my programming in bash, which is maybe even worse than doing it in R. And on the other hand, if I just put everything into the Python source file, then I have essentially 20 different files to do a time series plot. And now if my advisor wants me to change something about how I'm plotting my time series, I have to make 20 changes across 20 different files. So the compromise I eventually stumbled across was kind of making a general purpose Python script and then putting all my options in these YAML files. And I kind of landed on YAML because I liked it for its kind of Python-ness and indentation matters, and it's a little faster to type than JSON and about 1 million times faster to type than XML, so I was in a hurry. And basically this kind of tells the story of an experiment, at least if you are me and have been living in this world for several years. And it's basically showing the time points I want to plot from a spreadsheet that I manually typed numbers into, because in this case this is a plot of a experiment I did in the wet lab, so there's some manual process involved. I can't make the Python script transfer my cultures or move test tubes around yet. And then different treatment groups are just these different groups here. So I have a control and I have cultures that have been shifted into different light levels for different periods of time. And I'm just saying when I started that shock and when I ended, and that's just in the number of hours into the experiment, and the color I want it to be plotted with, and not shown here, but I just went back and kept adding more options for like which marker to use and line widths and all the options you can change in Matplotlib to make it look perfect. And the experiment this is describing was a pretty simple experiment in concept. I was talking about how these different strains tolerate light stress in different ways, and I kind of want to emphasize that even though these are photosynthetic organisms, or because they're photosynthetic organisms, they can actually poison themselves if you give them too much light energy at once. Partly because they have that tiny genome and don't have the defense mechanisms that like a more complicated plant would have. They just generate all these free reactive oxygen species, and those just react with the photosystem itself or with other cell mechanisms and destroy the cell from within if the cell doesn't have some mechanism of getting rid of or surviving that extra energy. And the story I really developed in the second half of my graduate career was how some of the strains were able to tolerate these changes in light intensity, and some were not able to tolerate it. So I did a lot of these experiments. I think I made probably hundreds of plots from them. So that's kind of the story behind a YAML file like this, and I, again, probably made 100 files like this and fed them all into one relatively smart Python script. And how that script works, I excerpted kind of the most fun part of it here, and that was just looping through the contents of that YAML structure, which turns into a nested dictionary when it's imported into Python. And basically for every group there's you saw there was a line for the condition that it was exposed to. And what this is basically doing is, with a lot of NumPy magic, going through the various rows of the spreadsheet and finding those that are relevant to what I wanted to plot for that group. And it comes out as a nice declarative statement of which rows I actually want. The right data are the ones that are the right time point, the right experiment, the right train, and the right condition. And then that just gives me all the data I need to give to the Matplotlib function on the last two lines. And yeah, that one's ugly too, so I'm going to have to drag it over because it's too important. Trackpad on this thing is actually as bad as anything else. And yeah, why that was showing up in the slideshow on one computer and not the other, I couldn't begin to tell you, but that's a question for, I was experimenting with making a presentation with Restructured Test after doing them in LaTeX and Beamer for a while, and it's easier, but apparently there's something about the way the PDF is saved that I need to look into. But the outcome is, again, this is the story I've been telling you about, is these 4 different treatment groups plus a control in gray are exposed to a much higher light intensity for 1 to 4 hours, and what you're seeing is the cell fluorescence, which is kind of a proxy for the health of the culture and for the number of cells in the long run, is actually plummets within minutes of being, or within an hour at least, of being exposed to that higher light intensity. And then after that shock, the ones that have been exposed for a longer period don't recover at all. And those that have been exposed for shorter periods bounce right back and are as good as new. And again, I did maybe 100 of these plots and emphasized different things and did it for different strains and different periods of time, and was able to crank those out pretty rapidly, at least once I had the data, because I had these scripts put together and had by then a pipeline figured out. So and again, just to talk about the versatility of the time series script, this is actually another input file for the same script, or maybe a later version of it. And in this case, the time series is actually the number of genomes you're adding to an experiment, and the result is the number of either unique genes or genes in common that you found. And that's what gives you that first plot I showed you. So this was that one that I did some by hand and some in R and some in Illustrator at the beginning of my career, and now I was able to regenerate it using a script that I pretty much had written with an entirely different purpose in mind, just by giving it a different set of input. And that's the story I really wanted to tell, is just how those little things like giving me the ability to organize everything on one page, and giving me the chance to fix font sizes and line widths and general visibility issues, and get things visible so I could push one button. And I told my advisor once that the dream was just to have one script that would run all the figure making scripts and then copy everything over into a LaTeX directory and compile my thesis and basically do everything except write it. And I didn't quite get that far. I think I had to copy it's had some bugs. I think I had to copy the image files by hand anyway, and never got around to writing a script to copy them over. So, and I had to write the thing, which was especially painful, but I got pretty close to that ideal, and that was all thanks to Matplotlib and John Hunter's contributions. So, when I was actually talking to Brian, I think a couple days before we heard the news about him, that I might want to talk about how I'd used Matplotlib. So when that news broke, it was pretty much time to go ahead and give this talk. And then, the more fun part of this talk is going to be harder to show you, because it was going to be more of a live demo. So
About 30% of the way through installing Matplotlib, in case necessary.
well, you'll have to set up Postgres too, so and import my import my database, and
There's definitely
but
ways for that.
yeah,
So you need to not be downloading all
yeah.
Torrents go.
Yeah, so I can at least tell you what this is about and show you a couple of images. So this was, I guess I started thinking about it late in my graduate career and played around with it a little bit, and since I graduated it's kind of been my hobby project, and every time I'd want to learn about a new technology, I'd fold it into this project. Like I actually had to spend time this week repairing all the damage I'd done because I was going to give it a big web interface and make it all jQueryized, and definitely leaving that part out for this talk. But the thing I was thinking about was this PITCHf data that I read about a few years ago, and this is kind of one of the big gee whiz things in the baseball sabermetric community. So if you've read the book or seen the movie Moneyball now, you know about this community of nerds that kind of talk about how baseball people were crunching numbers in the wrong way and how they could do it better. And people like Bill James didn't have the high-tech things like this, but they had plenty of data and went back and showed what really contributed to scoring runs and preventing runs, and threw some statistics at it that nobody had really thought rigorously about yet. And that was somewhat interesting to me, but also interesting was the new high-tech stuff. So this company has been putting these high-speed cameras in basically every major league ballpark now, so that when a pitcher releases a ball making his pitch, the ball is photographed like 100 times or something like that as it goes to home plate. And from that, they do a lot of image processing to find and report what the speed of the pitch was, both when it left his hand and when it arrived at the home plate, what its position was in the strike zone. And that's what this figure is all about, showing. This is actually a figure from the MLB website. If you've ever looked at the game day feature, it shows all the pitches as they're thrown now. It shows the direction the pitch broke and the amount that it broke. So a curveball generally breaks down and to the side, whereas a fastball stays up in the air a bit longer. And you can, in this picture, you can kind of see the curvature on each pitch, and you can see, I think the blue ones here are curveballs, and they're dropping a lot, quite a bit faster than the fastballs that are staying up or even out of the strike zone there. And it also, of course, they report the result of each pitch, like whether it was called a ball or a strike, or if the batter swung at it or put it in play or swung and missed. And from that, you can kind of have a lot of fun. So they actually put all this data up on a website. I think the story I've kind of heard alluded to secondhand is that they put it up there by accident. So things like smartphone apps could pull it down for drawing pictures like this, and then people started harvesting that data with their own scripts and putting it in their own databases, and baseball kind of said, well, it's out there now, so we'll keep doing that for at least this generation of data. So with all that, there are already these websites online like FanGraphs and Brooks Baseball that actually do a lot of plotting of this data. So if you wanted a real quick picture of what what people are kind of seeing with this stuff, you can go on those websites and get, like in this case, this is a heat map of the strike zone showing where the most pitches were thrown. But I had some issues with the way the graphs were kind of limited, and you couldn't make really complicated queries like search for only pitches in certain situations or that had certain results, and you were kind of limited to the power of whatever web interface they gave you, and they just had a relatively small number of drop-down boxes. Also, I had some minor complaints about just the way the plots were formatted, and having already done this all the way through grad school, I figured if anyone could make better plots, it would be me and Matplotlib. So some of the questions I had was, again, just could I do plots with different subsets of pictures and categorize pitches better, and so this could be a whole nother talk about just how I grab the data, and they're basically XML files on the website, so you need some XML parsing to get it into a database. It's obviously stored in a database on their end because it's all in a pretty well normalized schema and has primary keys and everything, so the importing is mostly just converting that into either SQL or SQL Alchemy arguments and putting that back into a database. And then have another script that's a SQL Alchemy client and uses a bunch of Matplotlib to make the plots that I want. And like I was saying, I was in the past month or so tearing up all the interface and thinking about how it would look on a website, and kind of setting out to learn more about Flask and web development, so kind of used that as my proving ground and got halfway there, and then kind of put all those features on hold when I went back to fix it up to talk about this week. And the examples, I should probably just copy them over from this one. So I'll just really quickly show you two plots because they kind of tell a fun story and they're fun to look at. One of these is, let's see here. This is basically showing not quite all the pitches he's thrown this year, but a large fraction of them. This is basically the profile of Chris Sale, who's the hero of the South Side this year, or one of them, because he's helping the White Sox stay competitive in the playoff race this year, where they were expected to go exactly nowhere. So he's a pretty hard-throwing left-handed pitcher, and what this is showing is basically from the catcher's eye looking at the pitcher where his different kinds of pitches are moving after he lets go of them. So for the most part, he throws these two kinds of fastballs, and they tend to run towards his throwing arm, and every pitch drops, but they don't drop as much as others because they have a lot of backspin, and that generates a little bit of pressure under the ball. Then he throws a bunch of changeups, which are, you can't see the speed in this plot, but they're slower and they drop more, and otherwise kind of look like fastballs out of a pitcher's hand. And he throws a bunch of sliders, which are a kind of a hard breaking pitch that move in the opposite direction relative to a fastball, and generally generate a lot of swings and misses if he throws it right, and he has a pretty good one. So he's a pretty conventional hard-throwing pitcher. And then there's, to contrast, is R.A. Dickey, who's a pitcher with the New York Mets, and he's basically been a fair to mediocre pitcher through his career, and it's been late in his career, and suddenly he's having this year where he's one of the best pitchers in the majors. And he comes at things entirely differently. He throws mostly a knuckleball, and the funny thing for most baseball fans know is knuckleballs are this mysterious, mystical pitch that nobody knows where it's going to go, including the pitcher. So he throws one, and sometimes it breaks right, and sometimes it breaks left, and sometimes it drops, and sometimes it doesn't. And it's a much softer pitch than pretty much anything Chris Sale would throw, but it forms this kind of big blob here that kind of represents that unpredictability where Sale's different types of pitches are a little better clustered together. So if you know Sale's next pitch is going to be a fastball, well, not you or me, but a professional baseball player would have a fair chance of hitting it. If you know Dickey's next pitch is going to be a knuckleball, you essentially know nothing because not even he knows what it's going to do after that. So yeah, and after getting all this environment working, I was kind of messing around with other plots and trying to dig into those questions, and I'm probably going to break it again when I go back and try to mix in some other technology. But it's kind of how I'm using Matplotlib for fun these days. Yeah?
Are there orange dots underneath the purple ones? Do you have any kind of transparency or a way to see overlapping data points?
Yeah, I should look into if there's a better way to do that. In this case, I'm pretty sure they're clustered up here, and there must be a few underneath. So one thing I did do, and I don't have those figures ready, but just tell it to leave the knuckleballs out of this picture and just draw the red ones, which are the fastballs that he occasionally mixes in, and just look at them separately on different plots. But yeah, this plot, I agree, is not perfect because it has that overlap and things are being drawn over each other. And that's another thing where I've been using Matplotlib a while, and it's probably got a way to do this better that I don't know about yet. Yeah? That's actually one thing that, well, that's part of the Major League Baseball data, so it's in the XML files you can download. And then people talk on the web about how their classification algorithm isn't the best, and like they'll mix up sliders with curveballs and change-ups and splitters. I'm not sure there's even a real difference, so it's kind of guessing at which is which if a pitcher claims to throw both of them.
Now, you're using polar coordinates here. Is that because the way the data is presented coming out of
the MLB site, or is there a reason for that? I mean, assuming we're looking at sort of the ball as it crosses the plane of the plate here.
Yeah.
So what's the advantage of the polar coordinates?
Yeah, the polar coordinates, they carve up the data in different ways. So in this case, I'd need to go back and look if I was calculating the break angle myself and then just applying the break length that they give you. But you can certainly plot it on XY coordinates as well. They'll give you the pitch movement in terms of just X and Y coordinates as well. So I've made plots in different ways and yeah, seen roughly the same story in this case. But yeah, they just give you the data as it's crossing a plane at the front of home plate. So yeah, if you had something that was kind of a backdoor pitch or something that came in from the side, because the strike zone by the rulebook is actually a three-dimensional area, then you might get misled by this data. But that's so much closer than the degree of accuracy that umpires achieve anyway that it's not really worth worrying about.
That's an umpire.
Yeah.
I prefer about that.
Yeah. Yeah, so that's all I had to show here, and just wanted to share something fun along with what I was doing for work for several years. And I'll probably be experimenting with it some more, and I've actually got it on GitHub, so I'll email a link to the list if people wanted to play with it. I've set it up on my laptop and on my desktop, so I've at least got it somewhat possible to actually port it onto a new machine, but I wouldn't guarantee that it's any fun. So that's all.
Question. Yeah. How real time is the data that comes through the baseball portal?
I'm not sure if they put it up as the game's in progress, but certainly by the next day. I mean, they're putting that data up for their that Flash app they have on their website and for smartphone apps and everything. So they're generating it in real time, and it's just a matter of when they make it available.
It typically becomes available overnight for some I know of, but it's like if they have their game tracker or whatever you can pay to get the real-time updates. That's not in stuff on the public feed. If I figure it out. It's the same data, it's just not public there.
Questions?
Are you still working with the bacteria?
I'm actually working with Brian at Textura now, so I was a little worn out with academia and wanted to do something purely software related for a change. So
Would you do it for fun, like in an open science hackerspace?
yeah, I'd seriously consider that. I think I'll have the Gitch for too long.
Oh, there's an open science meetup in public station on this
Yeah,
Plug.
yeah, the whole open science movement, open biology in particular, I'm very curious to see where it goes, because I mean, so much of what I did was kind of made possible by number cheaper sequencing, and it's the lab I'm still in touch with. They're at the point where they sequence the genome of a single cell without even culturing it or isolating it the way traditionally did. And I would imagine before too long it's going to be where you could just spend $100 and sequence like your sourdough starter or something and find out what's in there. and do all this stuff for finding out why your bread tastes the way it does.
Bacteria are like 40% of the production in the open ocean. Are they a big carbon sink in the whole carbon cycle? Or?
Yeah, they're a big part of it. And yeah, without them, you'd certainly have less production and less consumption of carbon dioxide and the ocean in general, not just the bacteria we studied, but there are photosynthetic eukaryotes, and they're a big part of it. That's not exactly why our lab got into it. We were more interested in just the pure science of knowing what's out there and understanding how their diversity and how their genetic diversity interacts with where they can grow, and that just teaches us things in general about the way these have evolved. But yeah, we've certainly made that pitch in the past, and will continue to. Yeah. The R Python interface, I actually heard about that around the same time as I was switching to Matplotlib, so never got around to doing much with that. But yeah, I've gone back to R and experimented with it some more, because there's just so many packages there if you want to do a specific statistical test that eventually I'll probably have something that I want to get from Python to R, and I'll have to look at that again.
Thanks.
Yeah.